Hello everyone, this is Sir Stephen, and as a part of medical microbiology, we will be looking at immunology, and the first series that we will talk about in this presentation will be your innate, general or non-specific host defense mechanism. The initial host defense mechanisms that sense and engage the potential pathogens are collectively referred to as your innate immune system, and it is an evolutionary older defense mechanism that relies on a limited repertoire of inborn sensor proteins that can provide an immediate response to pathogens without genetic rearrangements. And the innate immune system must be breached for an infection to occur. And even when unable to prevent or clear infections, innate mechanisms play essential roles in limiting replication and even directing the microbe-specific, long-lasting immune memory of your adaptive immune system, which we'll be talking about by the next video. The focus of this presentation is on the earliest events of your host and pathogen interaction. This host-pathogen interaction process and its general non-specific interaction will help us to understand how do they mitigate invasion and also the processes for detection and even possible clearance of your invading microorganism by the host. And if the infection is being established, a question like how do the initial interactions create an environment for the adaptive immune system to respond. And this particular table is coming from Mandel Douglas and Bennett's Principle and Practice of Infectious Diseases, and this will showcase the features of innate and adaptive immunity. So each of the rows will indicate you some of the features such as components, like for innate, you have physical and chemical barriers, mucus, AMPs, complement innate cells. For adaptive, that's humoral immunity, such as your B cells and antibodies, and cell-mediated immunity. For the receptors, they are fixed in the genome, and rearrangement is not necessary. For adaptive, they are encoded in gene segments, and we have to rearrange them. For recognition, this is non-specific. And for adaptive, it is very specific to a given pathogen. Responses includes chemokines, cytokines, phagocytosis, complement action, activation of effector cells. For adaptive, we have to activate, proliferate, differentiating of your antigen-specific B and T cells, secretion of antibodies, effector activity of your T cells, and the generation of memory cells. For initiation, for the innate immune system, there are a lot of immediate activation once it reaches the surface, mucosal surface, your skin, any of these organs, for minutes to hours. For adaptive, it could be primary response will be delayed, such as for coming days, okay? Beyond 24 hours, and rapid recall on subsequent exposure. So it needs to get acquainted with the previous interactions with that pathogen or that microbe. Duration for innate, it can persist for days, and contraction after elimination of pathogens for weeks for your adaptive. It could also be long-term memory for even months or years. This is a case of a probability of dying from an infection before a given age for adoptees with at least one biologic parent who died before age 50 years old, parent dead of an infection versus the adoptees whose biologic parents were alive at the age. So we have the parents alive. This shows that there could be an environmental or genetic factor that intervenes with this kind of graph. Your probability of dying with an infection is an accumulation of genetic and environmental influences. That's why this is the premature death on your adult adoptees was made possible. These were being studied of in the year 1988 for 148 families in that specific area. Moving forward, I have here photos of the epidemiologic studies, which will suggest that the BCG or vaccination with BCG at birth will significantly reduce the childhood mortality which caused by non-related infections. However, there are some evidences for this causal link is missing. And recently, a placebo-controlled trial showed that your BCG vaccination will protect you against an experimental infection with an attenuated yellow fever virus. This yellow fever virus vaccine strain that there are a lot of factors that will come into place, like your epigenetic, functional changes in your monocytes, which are indicative of trained immunity correlated with protection. Although non-inclusive, this provides us the evidence that trained immunity. So you have trained immunity will mediate at least part of the heterologous protection observed with your BCG vaccination. And there are a lot of studies over there demonstrating that there are some things we need to learn about the roles of your innate immunity that will play in protecting us from the invading pathogens. And in the Philippines, BCG vaccine was started in 1979 under the Expanded Program on Immunization Act. In this first section, we will talk about physical and chemical barriers to the entry of microorganisms into your body. We have here a histology of your sweat gland showing a stratified cuboidal epithelium. We take a look at the skin, the linings of the respiratory, urogenital, gastrointestinal tracts, which are comprising of thin layers of epithelial cells, and they were held together with tight junctions that form a barrier against the external environment. So this is kind of cell signaling properties of each of the cell following your epithelia. And the morphologic integrity of these surface is an effective first line of defense against your invading pathogens. But the epithelia functions more than that. Okay, they act more than just a physical barrier. As mentioned earlier, epithelial surface contain a lot of communities, a lot of microflora. They are commensal microbes, and they make antimicrobial substances, and they interact with their host cells to influence immune responses. So this is 40x sweat glands from scalp, a stratified cuboidal epithelium. So take a look at the morphology and the histology of each of these tissues. Your skin is forming a complex physical and immunologic barrier to invasion of your microbes. Again, as one of the largest organ of your body, the epidermis is composed mostly of keratinocytes, which are forming a physical barrier, and it has a key role detecting pathogens via a number of innate pathways, and it's not limited to one pathway alone. This is a section of your skin showing a large number of dendritic or Langerhans cells showing the epidermis in an M. ulcerans infection. This is using an immunoperoxidase stain, seeing photo on your right. And the epidermis also contains CD8 plus T cells and what we call as the DCs, Langerhans cells, that can sample of microbes, migrate to the draining lymph nodes, and prime the appropriate immune responses, showing you a desquamation of your skin or slowing of cells from the epidermis. This is a thick skin, human skin, of course. 100x at 35 millimeter. It shows you the epidermal layers, the stratum corneum, granulosum, and the spinosum, and at the base will be the basale, the dermis, the sweat gland ducts, and the desquamating cells on this portion that's slowing off the surface. There are several AMPs or antimicrobial peptides in your skin, which includes the beta defensins, the cathelicidin LL37, and the hexadecanoic acid, which are depressed on atopic skin, which is a condition where microbial superinfection is very common. And the relative dryness, salinity, and mild acidity, that's why we call it the acid mantle, because you have a pH level of 4.5 to 6 in your skin, combined with the normal skin microbiota, will help it make an inhospitable environment for your invading pathogens. Well, if you got a patient with some ulcerous lesions that occurred in someone with an inflammatory bowel syndrome, or it could be a fungal bacterial infection, they might be permeable and hence more hospitable to colonization. And this has been speculated that oily skin, the one that produces the sebum, a lipid-rich coating that protects and lubricates your hair and your skin, has antimicrobial properties. However, there are some bacterial species, including the Propionibacterium acnes, that hydrolyzes the triglycerides in the sebum, and they release the free fatty acids, and these fatty acids will ultimately promote colonization in your sebaceous glands. And the continual desquamation of your skin also aids in the elimination of the microorganisms. Showing you a photomicrograph of your P. acnes in a thioglycolate medium. And again, as what I've said, the continuous desquamation of your skin, the shedding of your skin, there are instances that it will also get rid of the microbes that may penetrate the skin, and they usually gain access by some physical manner, such as looking for an arthropod vector, it even be trauma, surgical incision, or intravenously placed catheters. With most of that, most pathogens enter the body through the mucosal surface, and these mucosal surfaces were found in your respiratory tract, your gastrointestinal tract, your genital tracts, and what we call the mucus will be forming by your highly glycosylated proteins called mucins that are often specific to the mucosal site, and they carry immune cells, antimicrobial factors, bacteria, nutrients, and even wastes. And the continual secretion of and elimination of your mucus will create a flow. This will create a flow that will require your pathogens to move upstream across epithelial surface. And together with the physical and the chemical properties of your mucus, it will create a robust barrier that is effective at immobilizing and inactivating your pathogens before they can contact to epithelial surfaces, while also supporting the natural inhabitants, your commensal microbiota, to subvert its function. Many pathogens have what we call as development of your strategies that penetrate, evade, degrade, and even disrupt the production of mucus. Mucous membranes. Showing you a photo on your right is lysozyme crystals stained blue with IZ dye. Most of your mucosal epithelial cells possessing the same peptide shield as those of your skin. However, there are some body secretions that include saliva, cervical mucus, prostatic fluid, and tears that are endowed with unique antimicrobial properties. For example, again, lysozyme, one of the most potent antimicrobial substances, which is produced by the intestine, your mouth, and even your eye. And this is very particularly effective against your gram-positive bacteria because it enzymatically degrades peptidoglycan, which is a minor component of a bacterial cell wall. However, many gram-positive and gram-negative bacteria have evolved their mechanisms to evade killing by lysozymes, in which if the cases, for an example, Staphylococcus aureus, impacting the balance. The S. aureus is impacting the balance of your commensal bacteria at the mucosal surface. Mucosal secretions by your mucous membranes contain very significant levels of immunoglobulins, which play an important role in the defense of these surfaces. And for example, this photo on your right is what we call as your SIGA, that is capable of neutralizing pathogens or toxins that are internalized into epithelial cells. And in addition to that, SIG-A coats the majority of your commensal bacteria in your gut. And this interaction will facilitate the targeting and sensing of commensals by tolerogenic dendritic cells in Pierce patches of your small intestine. It shapes the composition of your gut microbiota and contributes significantly to intestinal homeostasis. And as you all know, the gut microbiota is the most well-studied microflora in a human body. With its connection with the people's diet and lifestyle, it could detect what are some of the diseases that are attributed if there could be a dysbiosis in these particular sites. Again, secretory immunoglobulin A is the most abundant class in secretions other than those in the genitourinary tract and your lower respiratory tract, which has slightly higher levels of IgGs. Once a microbe reaches your alveolus, a physical expulsion it will be much less effective because unlike the respiratory epithelium, your alveolar surface is not ciliated. Okay? It lacks also mucus. And so, the reason for this is that it should be facilitating gas exchange. That's why it will be getting rid of this cilia and mucus to further the process. And as a result, alveolar epithelial cells rely on their intracellular defenses and stimulate the production of your type 1 IFNs or your interferons to prevent the further spread of infection. So your alveolus here forming a hollow cavity, which we call this little cavity, is non-ciliated and don't have mucus. Each breath we take, it brings us with a potential to inhale some of the microbes that can cause respiratory diseases. And if they are not eliminated quickly and reach the appropriate niche, okay, they will attack us. And to defend against this constant threat, the respiratory tract has a formidable array of antimicrobial defense mechanisms that must be compatible with the crucial functions of gas exchange. And immune defenses are coordinated in relation to your airway size. Okay? As effective defenses of the large airways, such as your thick mucus layer, can be harmful in smaller airways. The human trachea, bronchi, and bronchioles are lined by highly ciliated epithelial surface that continuously propels mucus upward and leading to the mechanical clearance of any trapped pathogens that are subsequently ingested. And this process, called coughing, is extremely efficient, leading to 90% of a deposited material being cleared in less than an hour. This is your respiratory tract pseudostratified columnar epithelia. And again, your inhaled microbes must first penetrate and survive the aerodynamic filtration system of the upper airway and your tracheobronchial tree. The same thing with your GI, your gastrointestinal tract. It must be able to sort through an enormous amount of antigenic exposure without harming the host. And although oral mucosa in this oral cavity has similarities with other mucosal sites, some of its immune mechanisms are unique. And as a first line of defense, your oral epithelium with the underlying connective tissue of your lamina propria provides a strong resistance to other shear forces of your mastication, and it helps prevent the penetration of microorganisms. And like the epidermis, the oral epithelium maintains its structural integrity and limits colonization by a process of continuous cell renewal to replace cells that are being shed from the surface. And secretion and accumulation of saliva will limit the forming of unwanted microbes. Saliva, on the other hand, carries a range of host defense molecules. They have mucins, lysozymes, AMPs, and Sig-A, and other immunoglobulins that are able to inactivate your pathogens. And unique to the oral mucosa, the periodontal epithelium around your teeth allows an access of serum proteins and immune cells, providing a link to mucosal immunity and systemic immunity. Showing you a photograph of an oral thrush, which is a common condition caused by the growth or the overgrowth of the fungus Candida albicans. So cases are characterized by the growth of matted yellow-white patches of fungus in the mouth. And one of the causative agents, your C. albicans, is leading to the candidiasis. Remember your GI tract, your stomach has a very highly acidic environment, and this high pH acidic environment of your stomach has an antibacterial effect with the use of, or with the aid of your pancreatic enzymes, biles, and intestinal secretions. Although they are effective, they are non-specific antimicrobial defense factors. Your GI tract also will take a note of this, that it has it must be coated in mucus that has different properties in your stomach, in your small intestine too, and in your colon. And in your small intestine, mucus limits the number of bacteria that can reach the epithelium and pee patches. In your large intestines, the inner mucus layer remains relatively free of bacteria, whereas the outer mucus layer support the growth of your commensal bacteria. This is a picture of Pierce patches, which are organized lymphoid nodules commonly found in the small intestines, and the histology section was taken from the ileum. So going back to mucus is continuously secreted from your goblet cells and your Paneth cells and move distally with peristaltic waves. The function of mucus is to expel potential pathogens and requiring bacteria to travel against this mucus flow to reach the tissue surface. And alteration of these physical and chemical parameters can lead to the increased susceptibility of the host to infection and chronic disease. For example, some of the evidence suggests that hypochlorhydric patients are more susceptible to pathogens such as your Vibrio cholera and Salmonella. And mice deficient for the most abundant mucin, MUC2, spontaneously develop colitis and predisposed to inflammation-induced colorectal cancer. Showing you the photo of a transmission electron microscope image of the V. cholera that has been negatively stained on your left, and yellow-colored sucrose fermenting colonies of your Vibrio cholera on TCBS agar. GI tract, and in addition to that, a number of disease states has been shown to be accompanied by the movement or translocation of gram-negative bacterial products across the epithelial barrier and into the circulation. And this is very often accompanied by loss of tight junctions. Okay, these loss of tight junctions happens between the enterocytes and or the loss of enterocytes altogether. And in some cases, it may be tied to loss or impairment of Th17 cells, a subset of T cells, which will be stimulating mucin and AMP production that improves tight junction function and also IgA transport into your lumen. Now we have to take a look at the male and female genitalia. Many immune mechanisms in the genital tract of both the men and the women bear similarity with other mucosal sites. And uniquely, showing you below, the female reproductive tract or the FRT must be able to accept a semo-allogenic fetus while also conferring protection against pathogens. Of course, it must be able to conceive the fetus which has the traits or the genes coming from the maternal and paternal sides, and at the same time protecting against the invasive pathogens. And to achieve this balance, we have sex hormones. They must be coordinating with the aspects of your epithelial, your stromal, and your immune cell number and function in each of the compartment through the rest of your menstrual cycles. In continuation, despite the complex defense mechanisms of your female reproductive tract, accumulating evidence will suggest that a window of vulnerability to infection may exist during the secretory phase of your menstrual cycle, which is programmed to function and facilitating fertilization. Multilayered squamous epithelial cells will cover your vagina and your ectocervix, whereas single-layer columnar epithelial cells will cover the endocervix, uterus, and your fallopian tubes. T cells are more abundant in your lower female reproductive tract, while granulocytes and natural killer cells are higher in the upper female reproductive tract. Your lumen of your FRT is bathed in fluid that differs between the upper and lower tract and across the menstrual cycle. Your vaginal microbiota also is very implicated in preventing a number of urogenital diseases, which includes bacterial vaginosis, yeast infections, urinary tract infections, and human immunodeficiency virus transmission. The human vaginal microbiota is dominated by 4 lactobacillus species. They are studied in most women of reproductive age, and they can be significantly having a difference in vaginal community composition among the women. And under hormonal influence, your vaginal epithelium contains increased amounts of glycogen that is depolymerized into simple sugars. Lactobacillus species metabolize these sugars into lactic acid and lowering the pH of your vagina to create an environment that will restrict the growth of most of your invading microorganisms. Your genital tract, you see in your left is a human urine, and urine was once considered sterile and is now known to contain a commensal bacteria. And although the contribution of these organisms to immune functions remains to be determined, we have the lower urinary tract, which is rinsed with urine 4 to 8 each times a day, eliminating the potential pathogenic organisms unless they are capable of firmly attaching to the epithelial cells of the urinary tract. In these cases, these pathogenic organisms are Neisseria gonorrhoeae and some certain strains of E. coli. You're familiar with urinary retention, right? Or lack of complete bladder. The lack of complete bladder emptying, it will impede the flushing process. Therefore, urine is bactericidal for some strains of bacteria, mostly because of its pH, although factors such as hypertonicity, urea, and the presence of antimicrobial peptides play a role. There is also what we call as the protein uromodulin, which is also known as the Tam-Horsfall protein, which is being produced by the kidneys. This is the most abundant protein in the normal urine. And in addition to that, protecting against your kidney stones. This uromodulin will bind to your uropathogenic E. coli, preventing these bacteria to form or gain a foothold on the cellular lining of your urinary tract. When it comes to the difference of your urogenital tract, the length of the male urethra is around 20 centimeters in an adult, and it provides a passive protection and bacteria which seldom gain access to the bladder in men unless introduced by instrumentation. Your female urethra is much shorter with 5 centimeters in an adult and is traversed more quickly or readily by microorganisms. That's why they're more susceptible with UTIs than in men. For the male urogenital tract, the external surface of the non-erect penis is covered with keratinized squamous epithelium, which is relatively resistant to infection unless the skin is broken or inflamed. In circumcised men, most of all of its foreskin epithelium is removed, leaving you a similar dry keratinized surface which is also resistant to infection. In uncircumcised men, the subpreputeal epithelia covering the inner foreskin and the glans corona or mucosal surface that are more susceptible to HIV and others viruses such as your HPV and your HSV. This is just showing you an anatomy of lacrimation, showing you the lacrimal glands, superior lacrimal punctum, lacrimal canals, lacrimal sac, inferior lacrimal canal, and the nasal lacrimal canals. And as what I have discussed for the previous few slides, your constant bathing of your eye by tears is an effective means of protection, which foreign substances are diluted continually and being washed away by the tear ducts into the nasal cavity. Tears also contain large amounts of lysozymes, SAGA, lactoferrin, and lipocalin. Now we move to the second part of this innate defenses with innate immune sensing of invading pathogens. I'd like to introduce to you such molecules, or we call the pattern recognition receptors, or the PRRs, which plays a crucial role in the proper function of your innate immune system. Well, PRRs are germline encoded host sensors. They detect molecules typical for the pathogens. They are proteins that are expressed mainly by the cells of your innate immune system, which such as your DCs, your macrophage, monocytes, neutrophils, and even epithelial cells. And to identify, there are two classes of these molecules. We have pathogen-associated molecular patterns, or PAMPs. They are associated with microbial pathogens, and damage-associated molecular patterns, the DAMPs, which are associated with the components of your host cells that are being released during cell damage or death. Your PRRs also is being called as a primitive pattern recognition. These receptors have evolved before other parts of your immune system, particularly before adaptive immunity. These PRRs also will mediate the initiation of your antigen-specific adaptive immune response and the release of your inflammatory cytokines. TLRs, or toll-like receptors. With this rapidly evolving field, there are several PRR families that has been described. TLRs, the best characterized PRR gene family, are expressed in both the innate immune cells, such as your macrophage, your DCs, as well as your non-immune cells, such as your epithelial cells and the fibroblast. There are 13 known mammal TLRs, 10 of which are found in humans. TLRs are generally classified into two categories, which depends on their cell localization, and PAMPs they recognize. Human plasma membrane-bound TLRs 1, 2, 4, 5, and 6 interact with microbial membrane components, whereas TLRs 3, 7, 8, and 9 are localized in intracellular vesicles where they recognize viral and microbial nucleic acids. TLR10 is the only human TLR member without known ligand specificity and function. Although recent studies suggest that distinct from other TLRs, it functions to suppress inflammation. PRR members of the CLR family are expressed as the transmembrane or soluble proteins which bind a complex array of carbohydrates, and they are very essential for the antifungal immunity. Microbial PAMs. There are several known PRR families again that also detect viruses or bacteria within the cell, and the NLR family, of which there are like more than 20 members in humans, will detect a range of microbial PAMs in your cytosol. RLR family, including RIG1, MDA5, are also called as the melanoma differentiation associated protein 5. The Laboratory of Genetics and Physiology 2 detects intracellular viral RNA. You can see in the photo on your right a scanning electron micrograph of a clump of Staphylococcus epidermidis in green in the ECM, which connects cell and tissues. Microbial pumps such as your cyclic guanosine monophosphate adenosine monophosphate synthase aim to, absent in melanoma 2, DNA-dependent activator of your IFN regulatory factors, DNA PK IFI16, and LRRF IPI or P1, which is leucine repeat flightless interacting protein 1, as an example. Although accumulating evidence suggests that DAMP recognition can mediate the aspects of innate and adaptive immunity, further research is required to better understand the extent to which the DAMPs can fully substitute for PAMs in initiating immune responses, or whether they are more often operate in combination with PAMs to elicit an immune response in the context of infection. Some PRRs are able to detect intracellular host-derived factors by cell injury or tissue damage, and we call them DAMPs. Specific DAMPs have been shown to mediate inflammation via the PRRs. I have an example here, the high mobility group mobile box 1 and heat shock proteins, which can stimulate the toll-like receptors 2 and 4. Showing you a photo of a colorized electron micrograph of a natural killer cell from a human donor from Innate. In addition to pattern recognition, your immune system has several strategies to recognize the absence of normal self molecules that can result upon infection. And the most studied example of missing self recognition involves your NK cells, or natural killer cells, a prototypical member of the ILC family. And NK cells are cytotoxic innate lymphocytes that circulate through blood and tissues to defend against microbial infection and the tumor progression. We have patients and animals with altered NK function are more susceptible to recurrent HSV, HPV, and varicella virus infections. And NK cells has specificity and functions which are tightly regulated by the complex balance of activating and inhibitory receptors on the cell surface. Activated NK cells also produce pro-inflammatory cytokines which can further stimulate your innate and adaptive immune responses. This diagram here will show you an engulfing of a pathogen by a phagocyte. It starts with a white blood cell, your phagocyte, Then we have here initiating receptors to detect pathogens. We have the viral proteins of your pathogen. Receptors will detect these viral proteins, attracting to a phagocyte. Then the phagocyte contains lysosomes that has digestive enzymes. They will become a phagosome. The phagosome develops around the pathogen, and lysosome attaches to and releases the digestive enzymes into your cell. Once it has finally reaches its purpose, the pathogen is broken down by enzymes into proteins and being engulfed by the phagosome. In this part, we'll take a look at the review of your basic cell biology or your basic biology courses as we talk about phagocytosis. Critical to the innate response are the cells that seek and engage invading pathogens. And as described earlier, both of your immune and non-immune cells at the barrier surface will express your PRRs that sense and trigger inflammatory responses. Next, specialized innate immune responses or cells with another essential function are the phagocytosis are being engaged. Phagocytosis is a complex mechanism by which relatively large particles greater than 0.5 micrometer, including altered cell particles, it could either be your necrotic or apoptotic cells, or invading microbes are engulfed within a plasma membrane envelope and then internalized into a large endocytic vesicle. We call it the phagosome. Your phagosome can then fuse with the lysosomes to form a phagolysosome, which will become acidified, and together with antimicrobial proteins and reactive superoxide and nitric oxide radicals usually kills the microbe. As what we have shown earlier, that diagram will explain a lot. Although many cell types are capable of phagocytosis, several including macrophage, monocytes, neutrophils, eosinophils, and dendritic cells are highly specialized for the process and hence termed professional phagocytes. Macrophages are the major phagocyte population in body tissues and have a diverse range of phenotypes and functions that in some cases are very specialized for the location in which they reside. Showing you a 3D rendering of a monocyte. Monocytes comprising approximately 10% of nucleated cells which are circulating in your blood. Although significant pools exist in your spleen and in your lungs, they can be mobilized when needed. And neutrophils, a type of granulocyte which is most abundant in the white blood cell in circulation, around 40% to 80%, provide a critical rapid defense against invading microbes. And in response to your inflammatory stimulus, neutrophils migrate into infected tissues where they bind, they engulf, and kill the microbes. And as also, they will release factors that are inactivating extracellular microbes and prime immune responses. In addition to enhanced phagocytic activity, each of these cell types are armed with a multi-layer of antimicrobial mechanisms that inactivate your pathogens, secrete cytokines, recruit immune cells, and prime adaptive immune responses. And we have two main functional types of dendritic cells. Okay, they are mediating into shaping adaptive immune responses. We have your conventional DCs and your plasmacytoid DCs. Professional phagocytes also variably express receptor systems that, together with the PRRs, they recognize microbes, particulate matter, and damage host cells. Receptors that facilitate your phagocytosis are classified as either your opsonic or non-opsonic receptors. And they initially contact a target with the phagocytic cells, resulting not only in chemical sampling but also physical assessment of the target through pseudopodias, membrane ruffling, and engagement of specific receptors. This is what's very interesting about the immune cells, because they have this chemical sampling ability to detect whether there are incoming pathogens in the bloodstream, where they can also trigger another cell or immune cell to respond to an immediate or incoming pathogen. And the physical and chemical properties of these targets will help us to instruct the nature of the subsequent response, which is also highly dependent on the state and function of your ingesting phagocytes. Many pathogens have evolved to interfere with different steps of the phagocytic process, but some, including Mycobacterium tuberculosis, Listeria monocytogenes, Legionella pneumophila, will survive and replicate in phagocytes. And these intracellular bacteria evolve unique strategies to perturb or evade phagosomal maturation and to withstand cellular antimicrobial factors. Autophagy, another mechanism, refers to self-eating, allowing a collection of processes that allowing cells to digest long-lived cytosolic proteins, lipids, and organelles and autophagosomes that fuse with lysosomes. Although autophagy primarily serves as a survival mechanism in response to stress or starvation, it is now known to mediate important roles in homeostasis, development, and immunity. And in concert with PRRs, other signaling pathways, autophagy can directly eliminate pathogens, regulate inflammation, secrete immune mediators, promote adaptive immune responses. It's through your antigen presentation on your major histocompatibility complex molecules. The importance of autophagy in immunity is being demonstrated by a range of mechanisms used by microorganisms to prevent, counteract, or even exploit aspects of the process to promote their survival. And for example, studies suggest that MTB survival in macrophage is in part due to its first, inhibit the induction of autophagy, second, disrupt autophagosome fusion with the lysosome, and to suppress autophagy-linked class 2 major histocompatibility class antigen presentation. And much remains to be learned about how autophagy will mediate your innate and adaptive immune responses that are also intrinsically linked to homeostasis. Polymorphisms in autophagy-associated genes are associated with increased risk of Crohn's disease. And emerging data also implicates that autophagy in other inflammatory disorders, metabolic conditions, and neurodegenerative diseases, and even cancers. Showing you here an aphthous ulcer on the mucous membranes of the mouth given the patient has Crohn's disease. And given its many important roles, there is a great interest in developing approaches to target autophagy pathway for potential new therapies and anti-infectives. Now we move on to the inflammatory response. The innate immune response is the assembly of signals arising from the range of PRR's pathways triggered by the invading pathogen, and the nature and magnitude of the response depends not only on the class of the invading microbe but also on factors such as anatomic location, level of replication, virulence, and host-specific variables. Of importance, immune defenses are usually balanced to effectively eliminate pathogens while also minimizing the damage produced by the immune system itself. When the physical and chemical barriers are being breached, the infectious agents enter tissues, a range of host factors and cell types are mobilized. Although many of the initial response proteins are always present, it is the rapid quantitative increase that constitute the inflammatory response. And upon sensing invading microbes or tissue damage, sentinel cells become activated and begin releasing small proteins called cytokines and chemokines. and other mediators such as your vasoactive amines, prostaglandins, will be a product of your complement activation. Here is an overview of an innate immune responses. Again, you will see here that mucosal surface comprise epithelial cells. They are interspersed with mucosecreting cells and other cell types that produce antimicrobial factors and sample luminal antigens. Mucus are carrying antimicrobial peptides, your AMPs, immunoglobulins such as secretory IgA, immune cells, other factors that can be trapped or inactivate pathogens before infection can occur. Commensal bacteria compete with invading pathogens and stimulate innate immune responses. We have also pattern recognition receptors on the surface. See here. And they initiate the signaling cascade that lead to the production of antimicrobial factors, cytokines, and your chemokines. Granulocytes and macrophage ingest and kill the pathogens and induce inflammation. DCs produce cytokines that activate your immune cells and signal tissues to resist infection. The inflammatory response enables the plasma proteins or the complement or the leukocytes to leave circulation and accumulate at the site of the infection. Moving forward, you can see here, once in tissues, they become activated, to eliminate invading microbes and infected cells. And if the pathogen is not eliminated, antigen-presenting cells, particularly the dendritic cells, will transport the antigen to lymphoid organs to promote adaptive immune responses. This is coming from Owen in the Immunology book, 7th edition figure. Another innate immune sensing mechanism is with the use of cytokines and chemotaxis. Cytokines are potent signaling proteins that orchestrate immune and inflammatory responses through communication between both immune and non-immune cells. In response to stimulus, secreted cytokines alter the function of target cells through binding to specific cell surface receptors. Cytokines can also act in an autocrine manner or autocrine pathway if the action is on the cell that secretes it, a paracrine manner if the action is on nearby cells, and an endocrine manner if the cytokine acts on distant cells. And although many have both pleiotropic and overlapping functions, cytokines are known to mediate many important roles during the innate immune responses, and they are very crucial to the transition to adaptive immunity. Detection of viral components also leading to the rapid secretion of type 1 IFNs, the alpha and beta, and other pro-inflammatory cytokines. Type 1 IFNs can be secreted by most of your cell types, although PDCs, which preferentially express the toll-like receptor 7 and 9, and constitutively express IFN regulatory factor 7, produce the largest amount of your IFN alpha or beta during a viral infection. Although best known for their potent ability to induce an antiviral state in both virally infected and bystander cells, your type 1 IFNs also have a roles in defending against bacteria and other pathogens. Other inflammatory cytokines are very important in early immune responses, which are including tumor necrosis factor, interleukins IL-1 and IL-6. Chemokines, on the other hand, are a special family of cytokines that attract leukocytes through chemotaxis to sites of pathogen invasion, and although some also have a direct antimicrobial activity and mediate roles in additional process. Chemokines are produced by macrophage and other cells after recognition of invading microbes or in response to pro-inflammatory cytokines. showing you an image of chemokines that are released by infected or damaged cells that forming a concentration gradient. Attractive cells move through the gradient towards the higher concentration of your chemokines. The interactions of your chemokines on target cell populations are extraordinarily complex. Although there appears to be a level of redundancy with multiple chemokines signaling through the same receptor, differential expression of chemokines, receptors, and adhesion molecules provides the appropriate instructions for leukocyte trafficking, and it is then the coordinated action of your chemokines. Other pro-inflammatory cytokines and vasoactive mediators that regulate the migration and activation of leukocytes in a stepwise process appropriately respond to an infection. And moving on to our next and last portion, this is the pathogen interference with innate immune responses. Essentially, all successful pathogenic bacteria, viruses, fungi, and protozoa have evolved ways to evade or suppress innate immune responses to facilitate their establishment and replication in the host. Major points of pathogen interference parallel three major steps in your innate immunity: PAMP recognition, intracellular signaling, and chemokine cytokine expression triggered by your activated transcription factors. First is pathogen interference. Altering the PAMP structure to avoid activation of PRRs is an important strategy in the establishment of infection for many pathogens and commensals. And for example, Helicobacter, Coxiella, Legionella, your Rhizobium have altered lipid A structures that are poorly recognized by your toll-like receptors 4. Several fungal species, including Candida and Pneumocystis, alter their surface glycans during different stages of life to prevent recognition by the CLRs, the predominant receptor family for sensing your fungi. Viruses such as your influenza A virus and dengue virus replicate in membranous cell components to avoid detection by intracellular RLRs, although others modify their viral genome. In the case of Crimean Congo hemorrhagic fever virus, or shield viral RNA through binding of viral or host proteins, such as your Ebola and IAV. In addition to avoiding activations of PRRs, bacteria also have developed multiple mechanisms to evade complement activation, including mimicry of complementary regulatory proteins and expressing the proteases, or small proteins that cleave or inhibit complement components. Here is an example of a secreted bacterial protease that may also act as an exotoxin, and be an example of a virulence factor in bacterial pathogenesis, for example, an exfoliative toxin. This is a bacterial exotoxic proteases which will be destroying some extracellular structures as aureus in their bacterial cells. This photo is a herpes simplex virus, and many pathogens express PAMPs that can be sensed by PRRs but evolve strategies to evade, and the most studied is the viruses such as your HSV. They directly inhibit the activation of PRRs, or target downstream signaling molecules, to interfere with the expression of type 1 IFNs or the function of antiviral effector proteins. Okay. The inhibition of this process is one of the mediated by virally encoded proteins that intervene in what we call as the post-translational modifications, such as phosphorylation and degrading key signaling components. One of the few examples of viral interference with innate immunity was the blockage of the proteins responsible for the antiviral effects of IFNs, and it is not now known that viruses such as your herpes virus family members also encode microRNAs. These microRNAs decrease the expression levels of specific mediators of TLR signaling, and the result is avoidance, of innate pathways, giving these viruses the upper hand. Bacteria can also have profound effects on innate signaling molecules or pathways. For example, the anthrax toxin subunit lethal factor targets the mitogen-activated protein kinase MAPK kinase for proteolytic degradation, which induces apoptosis of macrophage and dendritic cells exposed to this toxin. And finally, a third major point of pathogen interference is on the initial inflammatory response. For example, pox viruses have devised two ways of interfering with the activation of IL-1 beta through the NLR pathway. First, infected cells secrete a caspase inhibitor that prevents the maturation of pro-IL-1 beta. Second, the virus-infected cells also secrete a soluble IL-1 beta receptor, effectively reducing the serum concentration of these important cytokines. Taken together, these adaptations demonstrate the diverse approaches that the microbes take to successfully establish infection. In summary, the non-specific innate immune response provide the first line of defense that can prevent infections from gaining a solid foothold in the body. These defenses are described as non-specific because they do not target any specific pathogen. Rather, they defend against a wide range of potential pathogens. Thank you for watching, and up next, adaptive immunity, antibodies, and immunodeficiencies. Don't forget to like and subscribe to my channel. Have a nice day, and thank you.